Oh yeah, we had stopped here on types of antibodies, or the 5 types of immunoglobulins, or IGs. And I think we had gone through IgG, IgA, IgM, and the next one I want to talk about is IgE. IgE. So if you look here at these last 2 columns for IgE and IgD, we have way fewer of those that we make than the other types. And that's because these are for more, I guess, more specialized purposes. IgE kind of has 2 important, well, one important job in the body and one messed up job in the body. We use it to fight off worm infections, it's thought. So it's particularly good at helping to fight off worms that are infecting us. The other thing that it does that's not as helpful is that it is key for allergic reactions. So IgE is the antibody type that will help initiate allergy responses. And we're going to learn more about allergy responses next week. But those end up kind of harming us more than they help us. And IgE is very involved with that. So it's kind of thought that now that people have clean water and don't grow their own food as much and have generally better sanitation, we don't have as many worm infections. And maybe that's why people that live in more developed societies and in cleaner places have more allergies. Like there might be a link between those 2 things, but it's not completely shown yet that that's true. It's kind of a hypothesis. And I remember this because E, like ew for worms and ew for allergies. That's how I remember what IgE does. And then last we have IgD. IgD is different than these other ones because it doesn't float around. It stays stuck in the membrane of B cells, which are type of white blood cell that we're going to talk about next. So it doesn't float like these other antibodies. It stays in the membrane of a B cell, and it acts as an antigen receptor on the surface of a B cell. So that one, I'm going to draw it. Here's a B lymphocyte. It's got a big dark nucleus, and it's got out on its surface multiple IgDs that are acting as receptors to bind onto antigen shapes. So 5 classes. These antibody classes, the way that I ask that on an exam is they become multiple choice. So you can have up to 5 multiple choice questions about these antibody classes. Sometimes I'll ask about IgD. One other thing that I want you to go back and add about IgG in your notes that you had from Monday. It's very important to note that IgG is the only one of these that can cross from mom to fetus through the placenta. And I think I didn't have you write that on Monday. If you heard things like mom's getting vaccinated for COVID will help protect the baby when it's born, it's through this type of antibody, immunoglobulin G, that crosses through the placenta. That would help. And then if the mom is also breastfeeding, IgA would get transmitted to the child through breast milk too. So those two types are most important for protecting infants. Okay. So we make those types of antibodies and they help us in a variety of ways. And there are 5 classes of them. And we call the type of immunity that comes from antibodies humoral. And it's not humorous like funny, but humoral, which is fluid based. So like the plasma, or we have antibodies that are floating around in fluids like blood plasma and milk and body fluids, let's say. We call that humoral immunity. And this is coming from an old Latin word, the humors of the body, which is fluids. So they named the different humors or body fluids were blood, phlegm, which is like coughed up mucus, bile, got its own category, maybe urine was one of them. Different fluids of the body. So humoral immunity comes from B cells or B lymphocytes. And that's one type of acquired immunity that we have, is the humoral type that uses antibodies and uses B cells or B lymphocytes. There's another type of acquired immunity called cellular. And cellular immunity is going to use immune cells to directly attack. So it's not antibody based, it's cell based. In humoral, the antibodies are doing something helpful to attack. But in cellular immunity, it's cells doing the attack. And this is mostly the job of our T cells or T lymphocytes. Both of these cell types, B cells and T cells, they're versions of white blood cells. And they come from the lymphoid branch of white blood cells. So if we look back at our ancestry of blood cells, this set of cells that come from what we call the myeloid ancestry or myeloid lineage. I think we did this before. If we drew a circle around these are mostly involved in innate immunity. And all of the stem cells that we have in our blood that make all the kinds of blood cells, most of them end up something that's myeloid. The group that is lymphoid includes natural killer cells. They're kind of on their own sub-branch. And then we have the lymphocyte group here. So this group, this set of three is involved in acquired immunity. And if you've got this picture, I'm just adding to this a couple of useful facts about the lymphocytes that reflect what I was just talking about. So B lymphocytes, these are also known as B cells. T lymphocytes are also known as T cells. And they are going to give us the cellular style of immunity. T cells. B cells will produce something called a plasma cell, and plasma cells then produce antibodies. And together, B cells, plasma cells, and antibodies give us the humoral or the fluid-based immunity. This concept is going to come up in your nursing classes later on. And that reminds me, keep your notes from this class and use them in nursing school, because they're going to be talking about stuff that relates to this. And if you've got this to look back at it, oh yeah. They also become helpful for people who transfer here and didn't have the King versions of these classes. So help them out with these notes. So what we're going to look at the rest of today, and we'll probably go over a little bit into Friday also, is how are B cells and T cells and plasma cells giving us the acquired type of immunity. That's what's coming next. Okay, so B cells and T cells cooperate to together give us acquired or specific immune responses. And they're going to do that by having in their cell membrane, I'll have T right there. They're going to have in their cell membranes receptors. And so the receptor on a B cell is a IgD antibody. We kind of know what that looks like already. So this cell has this antibody sticking out of its surface, and it's waiting to grab something. And it's waiting to grab a specific antigen shape. On a T cell, they also have receptors, but we're just going to call those TCR for T cell receptor. And we're not going to get into their shape in this class. So when a specific antigen binds to the receptors on the surface of this cell, whatever the cell type is, it will get activated. And what we mean by activated is that when the cell binds its specific antigen shape, it is going to start dividing, kind of multiplying that type of cell. And it will also begin helpful actions. But they don't really start doing anything until they come across their specific antigen that binds their receptor. They wait until that happens. The other thing to note about this pretty wordy statement is they're not going to bind their specific antigen until the time when the person is exposed to it. There won't be activation of a certain B cell or T cell to do anything until the person is exposed to the shape that it binds. So I'm going to give a dumb example of that. So everybody hold up one hand, kind of like that. And we're going to imagine that all of us are B cells. And we all can bind something, but all of our hands have a little bit different shape and we bind something different. So like Isabella, hers is cup. Can you hold on to your cup? And Grace's is going to be bottle. And you could get your phone and you could grab your binder and you could grab your backpack. And mine is a marker for sure. And yours is that little brown thing on your keys. And I'm like, I'll leave you ladies alone. Okay, so put your item down, but hold your hand out. And we're gonna imagine that we're sitting around in the body, and we could be binding something, but it's not here yet, and we're just waiting. And then suddenly into the room floats a brown little pouch thing, and that's your thing. So could you like hold on to it? Okay, so her specific antigen came along. There's the brown pouch. And now if Madison was a B cell, there would suddenly be 1,000 more Madisons in this room, and all of them are ready to of that Madison, not that Madison, that Madison, she would like copy herself and make tons of Madisons who get ready to fight brown pouch because it's here. So we have lots of kinds of these, but there's only like 1 or 2 of them in the whole body. And they're not gonna start doing anything unless their antigen shows up. So like I could spend my whole white blood cell life waiting for a marker to show up and it never did. And so I don't have anything to do. Just waiting. First number never came. So I wasn't needed, but you just don't know. So our body makes so many kinds in case some shape shows up that we need to fight against. And so maybe today it's brown pouch and maybe next year it's plastic bottle and you get your chance. And there would be 10,000 Graces when we need plastic bottle fighting ability, but we don't right now. Today is a brown pouch infection. Okay, so once they get activated by the exposure to their specific antigen, they will multiply. So that would be the first thing that happens. They're going to multiply themselves, copy themselves, and that would come first. But then after they have multiplied themselves, this is what they would do. So B lymphocytes will produce a cell type called plasma cell. And the plasma cells be making a lot of them. And the plasma cells float in the blood plasma. That's how they get their name. And they are going to produce lots of floating antibody. And not just let's say antibodies, and not just any antibodies, but an antibody that is specific for that same antigen. So if this was Madison Blair and brown pouch came in her vicinity, we would get thousands of Madison Blairs, and they would make thousands of plasma cells, which would be making millions, if not billions, of antibodies that bind brown pouch. And maybe something that would help with that is if I try to draw this dude up here. Like, this is the angry antigen. That would bind the receptor and kick off this process. Has to be that specific antigen. And then we end up with a lot of antibodies that fight grumpy red tooth, whatever that is. So that's the B cell side of things. And we would make from that process IgEs, IgGs, IgA, I'm gonna leave it out, M. All those kinds of antibodies, but they would all bind that same shape. And the IgD is up here as the receptor on that B cell. So they don't float around, they're stuck in the B cell. At the same time, we would have same antigen, grumpy tooth there, would be able to also bind some T cell receptors. So those would bind and activate a T cell also. And the T cell is then going to multiply and end up producing a lot of other types of T cells called T helper, T cytotoxic, and T regulatory. And these are cell types, and they don't look really any different than the other cells on this page, but they're doing different things. All of them are white blood cells. So these groups of T cells, they each have a different job of what they're going to do. T helper cells' job is to assist B cells in all of their activation. That's why they're called helper. They help B cells get activated. T cytotoxic cells are going to assist in killing infected or dangerous human cells. Which kind of sounds like a natural killer cell, and it kind of is. They do similar stuff. But cytotoxic means cell killer. So T cytotoxic cells will assist us with killing cells that are virus or bacteria infected or cancerous. That's what they're supposed to be doing. T regulatory cells, I don't teach as much about in this class, but what they're doing is kind of quality control for the immune system. Like, don't overdo it, don't underdo it, do it just the right level. They're trying to regulate what the other cells do. And doing this stuff and dividing the cells and making the antibodies and all of this, these processes, all of this stuff takes time to implement. It doesn't happen right away because it depends on cell divisions and it depends on cell communication. So all of this takes time to implement. And that's why we say that acquired immunity is slow to develop. So it's slow, but once we get it, it's going to become strong. And this process, when it does get going, it's going to give us immunological memory. So there's one more cell type that I need to draw onto here that's not there yet. And it's going to be coming from here. So the other thing that happens when these cell types get activated by binding their specific antigen is they will make a cell type called memory cells. Memory cells. So there are B memory cells, and there are also T memory cells. And the memory cells are quite special. And we're going to write some separate things about those in a few minutes. So by making those memory cells, that's what it's going to give us, immunological memory. I'm going to leave that there. Pull the screen down. Come back to that. Look. Too fast. Too fast. Okay, so there's not actually a lot of new info coming up. It's reiterating that same info for the most part. Okay, so let's look next at versions of T cells that are drawn as cartoons. And if you've ever walked by my office door in between here and the bathroom, I have a poster from the person that draws these cartoons, and they're so good about showing immune system stuff in an understandable way. So this is a listing of types of lymphocytes, and it's actually got the versions of T cells and a B cell on there. And what I'm going to do on this is just tell you what is the name of each of these that we've already learned. So this is the B cell, and you can see on its surface, it's got antibodies sticking out there. And the B cell is responsible for making antibodies. So this is just a way to remember it. I'm an antibody maker, and it's a little bit smiley. Friendly antibody maker. The regulatory T cell is making sure that all the other immune cells do their job correctly, like a police officer. This one is what we have called so far a T helper cell. So it's called the other name is a CD4 T cell. This is a T helper cell. You could write that underneath here. And this one, CD8, that looks like an angry ninja, is the the T cytotoxic cell is this one. That's your major group of immune lymphocytes. T cytotoxic, the B cell, T regulatory, T helper. I want to go back to that previous picture now and draw in something. Draw in something. And if you're one of the people that have a lot of colorful pens, it's a good time to get those. So T cytotoxic cells have these proteins on their surface called CD8. And you can't be a T cytotoxic cell unless you have CD8s all over your surface. So some books and some teachers later on or some tests will associate those things together. So a CD8 cell is a T cytotoxic cell. A T helper cell is going to have all over its surface CD4s. And these are just proteins that stick out of their surface. Okay. So CD8 means T cytotoxic cell. CD4 means T helper cell. Okay, so all of these cells, all of these white blood cells or any type of blood cells, originally we build them in bone marrow. When I first started learning about anatomy and physiology, I thought that was weird. Like, why should our blood be made in the bone marrow? Thought that was weird, but I mean, I guess it has to be made somewhere. And the cells that make blood cells are so important to our body that you might as well put them inside of a hard protective surface and keep them safe. So they all come from the bone marrow. A B cell will finish its whole maturing process while it's still in the bone marrow, and that's why they're called B cells. A T cell will migrate from the bone marrow to an organ called thymus. So thymus is an immune system organ. It's right over top of our heart. And in little kids, it's really big because little kids are still forming their immune system. In older people, it shrinks the older you get because you already hopefully did that work of training your T cells in the thymus. So T cells finish their maturation in the thymus. And that conveniently matches with the T. T for thymus. So all of our cells in the human body, it's important for the immune system to know, is that a part of my body or is that not a part of my body? So we have on all cells of the human body some proteins that stick out of the surface called MHC, and that stands for major histocompatibility complex, MHC. And having an MHC class 1 is kind of like having a King ID. And probably, technically, all of us right now, I could get mine out of my office, ought to be able to pull out a King ID card if we needed to prove that we're allowed to be here. So everybody has that. Every human cell has this MHC class 1 that's sticking out of the surface. The only ones that don't have it is a red blood cell, but it's like, I belong here. I'm a part of this human's body. And these are examples of people that ought to have that. So the short hair version of Chase and the long hair version of Chase have, should have a King ID. And Ashley should, and Ryan Boer should, and this is a faculty member that's not here anymore. I bet she still has it somewhere. Even though she technically doesn't work here. So everybody ought to have that, and that's a proof of belonging to the body. So somewhere on that page, right? The proof of belonging to the human's body, having that protein. There are some cells in the body that have an additional type of protein sticking out of their surface called an MHC class 2. And this is telling you what type of cells have an MHC class 2. MHC class 2 is not like having a King ID, but it's kind of like having a security badge. And these, as of last year, these are the security officers at the Bristol, Tennessee city schools. Is the same one? These,
Officer Yuri.
is it this?
And Collie. I would see Collie more.
I see this guy more. I don't know. I see him at football games, but
I've seen the lady. I've seen all four. I've seen all four on the bottom. I've never
Yeah.
seen the ones on the top.
Yeah.
Officer Cade left Haynesville. He's on the Virginia side now.
Okay.
But officer,
These two walk around at the football games, I know.
yes.
And this one was the police officer at my younger boys' school, which I really like that. Like, they have a woman officer. That's
Yeah, everyone
Nice.
liked the ones at Tennessee High. I remember that.
Noah's his first week at Tennessee High, he got there late. And there were a bunch of kids in the front office area that were late, and one of them smelled like pot. And the police, like the police officers, could smell it too, and he was like, had to question all of them. And he started with Noah because Noah had long hair, and he looks kind of like shifty, I guess. It was his first week at Tennessee High. And it wasn't him, but it made a big impression. So if you're a human cell, you're definitely gonna have this. And if you also have some extra immune system security duties, you are also gonna have this, which is kind of like a police badge or a security badge. And that one is a proof. It's a proof that you have authority to enact defenses, let's say. And we're gonna stop there. Yeah, it's time to stop. Some immune cells have an extra identifier that, like, not only am I part of the body, but I also can do stuff that the rest of you cannot, because I am a special security cell. And I can carry weapons, and I can make dogs sniff people and stuff like that. So we'll have these slides for Friday, and then also the new set about vaccines, which is related to this also. See you then.
You teaching to next semester?
Not.
Oh,
It's gonna be on sabbatical. We don't know currently. The person that we were gonna get to teach it, they got a full-time job. And you like can't be like, no, you should, you should take this badly paying part-time job.
And I'm like, why?
It could end up being like if worst case scenario, it'd be professional,
I shouldn't say anything.
but we hope that he doesn't have to.
And so like, I'm walking.
Just because of his time and stuff like that, but it will
And I
happen.
just see all these officers standing right.
There'll be
there. I'm like,
somebody.
what's going on? And then I go in there and my mom comes in and my mom's crying. And I'm like, oh my God, I don't know what I've done. And then, Ashley should come in there because my mom and Ashley are best friends. And mom, they were like, mom, when my grandmother passed away. But it's, I was just like, well, I know, and Kim Kirk and Ashley, they just sat in there. As me and my mom cried. I was like, are you taking your test? Am I taking my test right now?
What time did you wanna take it?
Whenever. Good luck. When can I?
Do you wanna do it tomorrow?
Yeah, I can do it.
Okay, I have a couple of freshmen coming in for advising and then I was gonna leave
Okay.
after that, so
Yeah, that'll be good.
that would make things a little easier for me.
That'll be good. What time?
What time? Do you have a 9:40 class?
9:40, what is it? It's Thursday, yeah. I have a 9:40 and then I have I could come in after that class. If you could have it ready by then.
I mean, it's pretty much ready, I just have to get
Oh,
the
okay.
things out and,
And so 9:40 to 11:10, and then I think it's 11:10 to 2:40.
that you have open or,
Another class. So yeah, I have 9:40 to 11:10, and then I'm done until 1:10.
until 1:10.
Yeah, so 11:10 to 1:10, I'm free for 2 hours.
Yeah, come by, around 11:10.
Okay.
And I am meeting with someone at that time, but it's not like a long meeting.
Okay,
So that to me would be a good time.
that sounds good.
Yeah. And everybody was done in an hour or less
Okay.
yesterday, so you'd probably be done around noon. Something like that. That'll be good. I was thinking about that earlier, like, oh, we didn't say a time and.
Yeah, I thought about that earlier too, and I was like, maybe after class, yeah, but yeah,
I do, but I don't wanna rush you.
Yeah.
And then somebody's coming at
yeah, that's fine. Thank you.
2.
I'll see you tomorrow.